The overall goal of the following experiment is to use magnetic resonance imaging as a powerful tool to evaluate mixing in process equipment. This is achieved by combining two liquid streams in a split and recombined static mixer. MR images are obtained by selecting an appropriate imaging protocol. These images allow characterization of the mixer's performance. Results are obtained for an application relevant to personal care products, but the procedure can be applied to a broad range of food, chemical, biomass, and biological fluids.
The main advantage of using magnetic resonance imaging over other techniques such as video is that opaque materials can be visualized. Additionally, the information is quantitative in component concentrations and the degree of mixing can be calculated.
Visualizing mixing using MRI can be helpful for validating computational fluid dynamic simulations in manufacturing processes by detailed comparisons of spatially measured concentration distributions to calculated concentration distributions.
The SAR mixer is composed of a number of different plates laid into a PVC pipe. Each laser cut plate is composed of PMMA and cut 1.59 millimeters thick. Each plate has a rectangular key that aligns it along an acrylic rod in a PVC pipe. The plastic may be clear or opaque. The plates have various designs which have openings through which fluids can flow. The plates are laid into the pipe in a repeating pattern that results in tunnels that mix the two fluids that pass through the pipe. Plate S is used to stream the two fluids that enter the repeating motif. One fluid stream is at the center and the fluid streams above and below. They are at a relative flow rate of 10:1. Next, the fluids meet in an open channel which is made by 8 Type C